Hi, my name is Melena DeHilling, and I'll be presenting the effect of cardenolide concentration in milkweeds on monarch caterpillar growth. So my project studied the milkweed species Asclepias curasivica and monarch caterpillars. Milkweeds secrete a toxic compound called cardenolide as a herbivore deterrent, and monarch butterflies are highly resistant to this toxin. Therefore, their diet consists of the cardenolide-containing leaves. And as monarch caterpillars feed on the milkweed, the chemicals in the milkweed provide protection from the vertebrate predators. My main takeaway was that there was no significant differences between cardenolide concentration and the initial growth rate of monarch caterpillars. From the results, we can assume that cardenolide concentration does not primarily affect the initial growth of monarch caterpillars, and that the differences in mass are most likely due to the growth rate and developmental time. Other studies have shown that more cardenolide consumption actually increases sequestration of toxic cardenolides towards predators. My first figure is a scatter plot with both of the article's data. The x-axis is the foliar cardenolide in the milkweeds, and then my y-axis is the mass of the caterpillars. As we could see, there's a negative correlation instead of a positive one that we predicted. And the relationship between the cardenolide concentration and the caterpillar mass didn't have a strong correlation, indicated by the R value of 0.1906, which is less than 1. And we also ran a regression output where we could see that the p-value is 0.06, which is greater than 0.05, indicating that there's no significant differences between the amount of consumed cardenolide and caterpillar mass. As for my other figures, I did a scatter plot separately for each article's data. For the left one, I did one for day 5, which tested the masses of 2 monarch caterpillar species, Ecor and D. gillipus, and day 9 didn't have a specific caterpillar species that they tested on. As we could see the negative correlation as shown in the other figure, indicating that after consuming more cardenolide concentration, it has no effect. The importance of this study is to understand the complexities of plant and insect relationships, and the study of monarch butterflies and milkweeds is an important example of a mutualistic relationship. The monarch population will decrease without milkweed because it provides a source of food and a place for the butterflies to lay their larvae. And also there will be no pollination from the monarch butterflies, so there'll be less milkweed. Prior studies shown that the consumption of cardenolide-containing leaves affects the initial growth rate of monarch caterpillars. Therefore, it's important to study its effects on the monarch butterfly population. As my methods from paper 1, I used the plant species A. curasivica based on the total cardenolide concentration, and I disregarded the other species. And then on the right, I used the caterpillar mass for the same host species, A. curasivica, on day 9. And then for paper 2, I use the foliar cardenolide content with A. curasivica on caterpillar mass of 3 species, Ecor, D. gillipus, and D. pelixpus on day 5, so which was kind of different from day 9. And that's all. Thank you.